it's my pleasure to introduce the next speaker, Corina Brussaard, who's a senior research scientist at NIOZ, which is the Royal Netherlands Institute of Sea Research, and she also holds a special chair at the University of Amsterdam. And she's actually one of the leading scientists on Antarctic viral ecology. And today she's going to talk about the ecological importance of marine viruses.
Thank you. Thank you as well for the invitation to talk here. I really, really appreciate that, and it's an honor to talk after you, so thank you for that. Today is not about Antarctica. I'm sorry for those who came for polar research. There's no slide, but I will tell you we're very busy with trying to find out who infects who. Today I'd like to talk to you about the ecological importance of marine viruses, and a few things have been said. I will focus mainly on lytic viruses, and those are viruses that kill the host. So they enter the host, they actually have the host go and propagate for new viruses, and they come out. So the more lysogenic viruses, those are the viruses that build into the host genome for a certain period of time, and with a trigger they also go into a lytic state. I will not address, but those are especially virals. It's a life strategy that is found for prokaryotes, and also Prochlorococcus is facing that and has benefit from it as well. So today I just thought, I wasn't sure how much we would have about viruses, so we just go briefly through. We know our virus on our computer, but what's now the biological virus? Because it's interesting. I mean, they're bundles of genetic material. We all know that from textbook, and they have a little capsid of proteins and sometimes some lipids membrane, and they are obligate parasites. And the third point is really where it comes into a discussion. We call it the most successful life form. They're not alive, but they're definitely a life form if you ask me, and other people as well. And we have had a think tank meeting at some point, and our assignment was, what does the world look when there are no viruses? Yes, and what do you think we came out with? The viruses would have evolved even in that world, because it's too stupid not to do. It's a super successful way of propagating genes. They're very diverse in shape and size. Used to be about 200 nanometers max. We're finding bigger and bigger viruses. They're called these giant viruses. And I also have a slide on that. And they also have very interesting shapes, as you can see here. We're used to think about viruses like this, and maybe just a hexagonal, a no-tail. But we have some really funny ones here. And sometimes it's hard to actually recognize them under electron microscopy. So now we're going to look at the marine biological viruses. And yes, they're the same here, but there is this ocean full of discoveries. It's not just a new virus for another host. It's actually really new viruses. It's a tremendously interesting period to try and find out, what kinds of systems we run. We heard a little bit about the giant viruses here. Look at them. They're 1.2 to 2.5 megabase pairs. Prochlorococcus is 1.7. There you go. Outcompeted. Look at the size here, 1 micrometer. We still call it a virus. It is actually a virus. And here we have this only 460 kb. For some time, I had the biggest virus, but it was quickly, outdated. But this is a Phaeocystis. This is an algal cell. So here we see chloroplast and the nucleus and the onset of the flagella. And this is after infection very quickly. And the interesting thing is, all these little dots are the viruses. If you stain this with a nucleic acid stain, this is called a virus factory. So you don't see this nucleus of the host anymore. So if you're alien life, you go to the Earth, of course you bump into the ocean, and then of course you bump into some kind of microbe. And if you see this, you think that this is the host DNA. It's in the chloroplast, and it's huge. It looks like a nucleus when you actually stain this. And why is it called a giant virus when it's only so small? And that's because it shares these core genes that we have now described for giant viruses. For example, this translation genes, the tRNA, is found in all of them. And this is also the time when we have discussions really opened again. What is earlier, the chicken egg? Was viruses first? Or was life first? This is a heterotrophic, these are Microbes viruses. We have a heterotrophic nanoflagellate viruses. We have now some algal viruses that have giant viruses in them. And they all have pretty similar of these so-called giant core genes. So was this an ancestral organism that lost its organism function? Or did they just did they gain by mobile elements they gain, but then they gained all the same information. So it's a really interesting time again for having discussion on evolution. I'd like to talk about ecology at this point, but you can fill easily an hour on this one. The other thing is, briefly said, there are these phaeophages. They're the cool systems that we have viruses infecting a virus, and it's beneficial for the host. By killing the virus, by reducing propagation of the big virus, there is less big viruses spread out, that they are less infecting. So the epidemic feature, in a way, is reduced. So it's a very relevant thing for a host to have a phaeophage in you, right? Because it's not directed necessarily to you. So this is also interesting, and it's really proven to be a phaeophage. Not sometimes you need two viruses to get the whole system going. Now, in this case, it's really a function for the big virus. So let's just go and look at ecology. This is the no-brainer in a way for us, but for lots of people, it's not. We're used to look at the Earth like this. Turn it around, and it's all water. So if we are looking at viral ecology, yes, and I want to also blow you away with numbers. Look at it, 10 to the 31. Yes, we won, right? But there are more viruses than hosts, so it's logical if you have a lot of hosts that you have a lot of viruses. But the oceans are deep, and there's a 71% surface, so you have automatically a lot of viruses. Great, cool. We want to know, who are they? That's what there's a lot happening at the moment. A lot of metagenomic analysis. Who are these viruses? What are they? The next question is, what are they doing? And this viral spec, yes, this is my Antarctic research at the moment down there. But not all of them are infective. Not all of them infect at the same time. But per second, we have about 10 to the power 23 virus infections taking place in this global ocean. And we just for the fun, together with Catherine Legrand, I just yesterday did the math really quickly. That means if all the viruses would infect all the Prochlorococcus, so they will be all very selective for Prochlorococcus, it takes you 3 hours to wipe out the global Prochlorococcus population. So luckily, they're not. Viruses are very, very selective typically. Not all of them, but typically they are. So we would like to know a bit more. What happens when you have these lytic viruses? Okay, we have these populations dying. What does that mean? So who do they actually infect? Because we are so prone to think about, copepods, and there's an orca down here. But actually, when you look at the oceans, right, this is like the same with an iceberg. Most of the biomass of the iceberg is underwater. We can't see it. The same thing, in a way, most of the living biomass in the oceans is invisible, right, is microbial. So more than 70% is microbial. And since these are unicellular organisms, they are with many, many. And being with many is handy for a virus because your meeting chance increases. Right? So there's a lot of these ones are actually infecting microbes. So what happens when you are infected? In this case, it's an algal culture. We have particulate organic matter here versus time. And the dotted line, we started to infect the culture. So the control keeps growing and then goes in stationary phase. But here we infected them. It takes a bit of time to make new viruses. And then they all come out, these viruses, and then the culture lysis. Now, if we look here at dissolved organic carbon, and this is a ratio of labile versus refractory carbon, we can see that the control has some excretion going out, but especially viral infected cells, that's where these ratios steeply increase. So what the material that comes out as cellular material is actually quite labile, is easily biodegradable, and that's what thrives a lot of the bacteria. So this brings us to this viral shunt idea. So you kill phytoplankton and you kill everything, but you end up here in this dead cells dissolved organic matter pool. So you're really shunting material, particulate organic matter, away from the higher trophic levels to this microbial food web, which for Prochlorococcus, in a way, is very paradoxical, very interesting, because during that process, you get nitrogen, phosphorus, iron all back into the surface ocean, because they're tiny little cells with tiny little corpses. So they don't sink out right away. They're recycled in this upper ocean, bringing back nutrients in the upper ocean, which helps the phytoplankton that are limited in their growth. And we have also discovered, in a way, like a new pathway, because as I tell you, this is all when the cell lysis. So we got infection, takes a while in the cell lysis. We've done a study using nanosims and stable isotopes, where we actually show that the cell loses up to 30 to 50% of its cellular carbon and a little bit of nitrogen within seriously a couple of hours after infection, while the lysis only takes place after 12 hours to 24 hours. So we have a whole new leakage pathway that drains the cell from a lot of its carbon while making new viruses. So this viral shunt is actually speeded up even. And that same organism, that in the same study, this is a nanosym picture, we see some really interesting features here. These are called star-like structures, you understand why. And these are very, very long spines of chitin. This is the structure rolled up in the cell in a viscous form when it's still in the cell. Then it is excreted out, pushed out, it opens, it unfolds, and it hardens into chitin. It's not really clear why it's for, and the most logical or the best defendable hypothesis is that it is protection against grazing. Now, if we have non-infected cultures, we have about 36% of the total cellular biomass being excreted as cellular star-like structures. Quite a bit. I mean, there must be a function for this, right? An important function. Now, if we actually have infection, we don't see this excretion of these structures anymore. And that, if this hypothesis is right, so if you're an uninfected culture, you've got a lot of these structures, and you might actually prevent or reduce grazing. Now you're infected, you're not excreting these structures anymore, that could potentially stimulate grazing on infected cells. Right, so we've got the viral shunt where it's all logical, you go that way or you go that way, right? And now I'm saying, you might actually also make connections here. You might actually be able to increase as a host cell through your physiological state and morphotypes. You may actually be able to manipulate the system a bit and stimulate grazing on infected cells. It's just to complicate everything a bit. That same system is a really interesting one to actually study the viral control. Viral control, typically, as we read in textbooks, we talk about reduction. Something is increasing, and it just doesn't get as high as it would without viruses. At some point, the viruses take over and the population crashes, right? It's reduction. If you have utter control, it would never bloom. You have a situation where you actually are able to prevent the bloom from the start. And this is a situation which you find a lot of the abundant species. They're out there, but you never see them bloom. It's Micromonas, for example. Prochlorococcus is a good example. And one of the things in there is also that it really enhances stability of the system. And also, if you look at Prochlorococcus, and Penny really showed very nicely, that's a huge diversity. So if we think about this, it might well be that we have all these tiny little strains going up and down, but overall it looks like a steady, constant abundance. Standing stock doesn't really change. There's a lot happening within, but this is prevention of bloom. It's utter control. And this is a situation that happens, if you ask me, a lot in the oceans. So this is just the example of this Phaeocystis, so the single cells with or without star-like structures, but it can actually make big colonies. So over time we see a bloom of colonies and we see a crash of colonies. Colonial cells have a harder time to get the nutrients in because they have this matrix around them. And so when the nutrients become depleted, they will shed their cells. And you see a steep increase in single cells, partly because they are all shed from the colonies. And at the same time, you see a steep increase in the specific virus that goes along with it. Handy to use flow cytometry. We developed a method to actually account these little buggers, these viruses, and especially this one shows up with its own population, so it's a really nice model system to work with. So this is an example of reduction. I hope we all agree. Now, it's the same plot in a way, the same colonies. What we did until day 11, we didn't give it a lot of light. So we actually blocked the photosynthetic access. The cells could grow, single cell they could grow well, but they didn't have access photosynthetic material to excrete and to make a colonial matrix. And after day 11, we put on the lights, and then they could, but they didn't. We only have a few. And they're very tiny, because these ones actually go up to 10,000 cells in a colony. Here we have 16 at max. And, oh, then we must have a lot of single cells. No. But they can grow, because they can just handle that lower light. Doesn't really happen either. And now the same the red one is the former plot, but then with a rescaled, we can see that we actually have a buildup of viruses here. So this is really a situation where the reduced light in the beginning reduced the colony formation. Single cells could still grow, but they were very readily infected, so they were building up slowly viral stock. And at the same time, when we switched on the light and we could potentially have colonies, there were just hardly any cells to make colonies because the viruses were on top of them. So this is really an example of prevention. And it's all triggered by a growth-related factor, light in this case. It induces on the cell morphotype. So if we look now at a host physiology manipulating these interactions and the success of viral infection, we have here burst size. Burst size is the number of viruses produced per lysed host cell that comes out, and we have 3 different light conditions: high light, mid light, and low light. We look at the replete conditions, so phosphorus nutrients are plenty. We see that we have at the mid light quite a high burst size, right? A high production of viruses. We have low light, we have limitation of energy, we get a reduced number of viruses produced. It takes longer and less viruses. Now we have high light, this is actually an example of inhibition. It's also not really favorable. So we also see reduction, and it also affects the length, the period of making new viruses. If we put them now under P limitation, the red version, we actually, there's no effect of light anymore. It looks like P limitation is the only most important factor. But I'm saying here that there's a synergistic effect. That doesn't look like a synergistic effect to me. However, if you look closer, and this is where it comes in, we looked at the infectivity of the progeny viruses. And in both cases, we had a tremendous loss of infectivity. So these viruses were produced, but the combination of a light limitation with a phosphorus limitation just, well, we'll make them, but I don't know whether they work. Right? That is actually going on. So this is really important to study these co-effects, as I call them. Don't just look at one factor in the ocean. Look at multiple. They have a different way of integrating on the system. Then, since we are focusing on Prochlorococcus, this is an environment with lots of oligotrophy. That's very nutrient poor. And it can actually be interesting for an algal system to be in a system like that. Under replete conditions, sorry for the blurred part, didn't really see that. So under the replete condition, you have this burst size of about 300 plus. And now we have them growing, what they call a continuous culture. They're really adapted to a certain, you put in the medium, there's the culture, and you take out the cells. So the cells that want to stay in this pot need to grow with the speed that you put the medium through. So you can really regulate their physiological state, all at the same growth rate. We did that, and they grew near Mumax, so 97% of the maximum growth rate possible in a replete condition here on this red, or a third of the maximum growth. You can see that that immediately affects the production of the viruses. But now think about it. If you can handle a phosphorus-limited state, you can grow near Mumax, like for example Prochlorococcus can, and you have a tremendous effect on reducing the virus load. That's a pretty cool state to be in. That's actually like a refuge being there, right? Yeah, we get the thumbs up from Fanny here. So this is an interesting thing to think about. It's not all bad that it's low for the host. But then securing that niche has a risk. We gave them hourly inorganic phosphorus additions to mimic recycling. We saw that there's a little bit higher production. But now we gave them also organic compounds. And we see that they use, and this is an axenic culture of micromonas, and it readily uses this organic phosphorus source. So axenic means there's nothing else, just that algae. We have efficient utilization going on here. And that's awesome for the host, because there's another way of finding your phosphorus and stay in that niche. But it comes with a risk, because when is the organic phosphorus being released? When the cells, for example, lyse. So you got cells lysing, and the surrounding non-lysing cells think, hey, that's cool, that's organic phosphorus, I can grow better. But by doing that, they also have a higher load of viruses they produce, which actually induces, again, lysis. And that's great, but you see, there is this positive loop in a way there. So it comes with a certain risk, probably a balancing issue. Then I would like to take this, because there's all these physiological parameters that control the host and sometimes also directly the infectivity of the viruses, like temperature does. They are all very important when you look at global climate change. And for the North Atlantic, there is a prediction really that we have a more strengthened vertical stratification, which means that with warmer water on top, the water column stabilizes sooner and stronger. And that means less mixing from below, less nutrient inputs into the top, and so the system runs on regeneration. So we get smaller cells, we get a little bit less primary production, but we really wanted to know, and we wanted to predict, what is happening for these temperate regions when we have a situation with global climate change. You can wait 50 years in one spot, but there's no funding agency who wants to fund you. Maybe you can use the money for that, just Penny. But in general, we try to do a gradient and interpolate results, and that's what we've done. So we looked here at the gradients from the Canary Islands to Iceland, and to see if we want to predict what happens in the temperate regions, we are looking at the subtropical regions. We see over the transects from north to south, from south to north, my apology. We really see that we have a nice vertical stratification going on that's slightly weakened. It is slightly weakened towards the north, and we also see that we have a tremendously low phosphorus in these stratified regions, and it also holds for nitrogen, by the way. Slightly relaxed in the north, again. And you can see very well, this is what they call the deep chlorophyll maximum. It's for these cells interesting to have light, but they also want nutrients. For nutrients, you have to go down. There's maybe a little bit of mixing, but there's hardly any light. And for light, you have to go up, and then there are no nutrients. So you get this, like, you know, this in a way, suboptimal area, which you call the deep chlorophyll maximum, and lots of cells have developed to really be able to grow there. For example, Prochlorococcus low light ecotype. Tiny cells, and then here, with a little bit more relaxed circumstances, we see larger cells popping up as well. We looked at all these different cells, and we wanted to know, we optimized the method that was existing, and we wanted to know, we want to look at the grazing, and at the same time at the viral lysis rates. So is viral lysis happening? Because we keep telling you that from cultures. Perfect, but is it happening in the field? Is it really important? These are just 5 of the 8 groups we have, and there is also Prochlorococcus in there. And here you can see in blue is the grazing rate per day, so the specific loss rates, and in red we see the viral lysis rates. And I think I can say very quickly here that I hope you agree with me, viral lysis happens all over the place. And it's not really, doesn't seem to be really, you know, like only from one specific example. It's quite variable. Sometimes it takes over the total loss, and sometimes it's hardly in a viral lysis. But if you average it, and this is 2 cruises, by the way, average. If you average, we have Synergistic, the Prochlorococcus low light, because we focused on a deep chlorophyll maximum, because then we have some cells to count. And we put all the pico-eukaryotes together and all the nano-eukaryotes together. You actually see that viral lysis is equally important as grazing. And that's really important. It's way more important than we thought, because originally, traditionally, this is how ecosystem models run. You have gross growth rate or production, and we have grazing. Sometimes we had sinking, you know, sedimentation. But it means in our case, and with our data, that we have here, this is gross growth rate, this is grazing, and it's just economy. We would have a net growth rate which is surplus. We would, according to this data, we need to see an increase in standing stock, which you don't in these oligotrophic regions. And now with the measurements that we did, we actually have, this is an average viral lysis, and add them 2 up, together, you actually see that we have a match. And this is really important. We really should start putting viral lysis in our ecosystem models, not just to compensate for the total production, but also because it's a very different pathway of where the matter is going. So when you plot them for all the different groups here, the total loss versus the gross growth rate, you see this nice, for us, this is a really nice one-to-one line, but it means that viral lysis is important to explain this high turnover in these oligotrophic regions. And we are working on a data set now that is from a spring bloom on the other side of the North Atlantic, and that doesn't work anymore, because it's spring bloom, actually, you have a net increase. So it's cool, it's not an artifact, it's seriously business here. So an oligotrophic ocean, we can say what is produced per day is lost per day. It's not that there's nothing happening. There's a lot happening. Look at the growth rates which we had. They are high. And also Penny already convinced you that Prochlorococcus is a high growth rate. It's just that whatever is produced is also lost. So for us, it looks like a desert, but it's not. So this is the slide that I want to get to. Here is where we put viral lysis versus grazing. So above the one-to-one line, it's a virus-dominated ecosystem. Below the one-to-one line, it's a grazing-dominated ecosystem. So if we want to predict, and that was our original goal, what is going to happen for the temperate regions when there is warming, we will see a shift from a grazing-dominated system towards a viral lysis-dominated system, with all the consequences of it, right? I didn't want to put it clearly a question mark. It's not my expertise, but if you just follow the reasoning, you can have a more regenerative system. And that potentially also because there is less nutrients, we will have smaller algae already, we will have a reduced primary production already. That's without the viruses. But now with the viruses, that system is even going to be more regenerative. That automatically means we potentially have less fish. So I think, again, I hope I convinced you here that these viruses are really important to take along in our more global perspective. So that brings me to my last slide. I think it's really imperative drivers of biodiversity and coexistence. I didn't really talk a lot about it as such, but they really are driving the species succession, for example, with reduction control. But they also really increase the coexistence when they are more on a prevention level. Because if you are diverse, if you can resist a certain virus, you can actually sustain that. You may have a competitive disadvantage, but on the other hand, you stay alive. So you just take a little bit longer to get your thing done. And if you have that big mix, and for example with Micromonas and also with Prochlorococcus, that is the fact. You see, they are you can easily find for Micromonas a virus. But there's just a lot of different ones out there, and there's a lot of different Micromonas strain. And some of them are infected by one virus, and the other ones are not. So it's very, very diverse, and that's also how they actually promote coexistence. And of course, they are also important for the genetic biodiversity if we talk about the lysogenic, the temperate phage. The important mortality agents, their activity controls the matter and energy flow, and that's really why we should put them into the ecosystem models. Environmental variables regulate the host morphotype, the host physiology, and as such also the virus proliferation. It also may impact directly virus infectivity. We've done some Arctic studies where we show that the host is very sensitive to temperature, but it's super interesting to find that we have with the 5 or 6 viruses that we tested, that there are viruses that have the optimum infectivity at 0 degrees, that there are ones that cannot be frozen to -20, that there are ones that love it to be at -20, so that could be a nice virus. And when you increase the temperature just from the 3 to 7 degrees, which are the predictions, you see that some virus strains just completely lose their infectivity. So we'll also see a shift in who is infecting who with time. We have the global climate change alters the share of the viral lysis, at least for the North Atlantic, and I'm pretty sure it will also happen in Antarctica, but those data are not out yet. And I think in a way that's, sometimes people complain in the labs like, oh, another complicating factor. But it's really relevant, and it's the same puzzle that Penny has, right? You dig deeper, and the same thing in our lab. We just wait, and then there's another question popping up, and it's also very motivational. And with that, I'd like to thank the people in my lab who have done most of the research, and also spent time on Antarctica, for example, 2 times 5 months, which is quite a challenge. We've done a temporal there. And I also thank the collaborators in the funding agency.
Excellent. I'm scared.
They're very selective. You just need to know where you swim.
Okay. We'll take questions, please. Don.
Well, thanks for a great talk. A quick question. So if I get you right, in the oligotrophic sea, the daily production is balanced by grazing and by viral infection. But what about export production?
Well, so what you have here is up to 20 microns, and the host cells. And for especially the below 10 microns, the export production isn't very big. At least we didn't find it to be very big. Recycling is really occurring in the upper water column. There is the potential, of course, of forming aggregates, and yeah, something happens. I mean, there is a biological pump even in the very oligotrophic region, so stuff is going down, but it's not the majority of the cells. So I'm talking more here about the bulk. There's a lot of recycling going up directly there. And the viruses are contributing to that, to keep that in the upper water column.
Thank you for an excellent talk. I'm not sure I understood this correctly, but did you say that when Phaeocystis was in its colonial form, that it was somehow protected against a viral infection?
Yeah, sorry, I should maybe have said that a bit better. There is a hypothesis that the cells are infected, less infected, protected, thank you, because you've got the matrix, right? So you have to go through the mucus. That's one. But there are viruses that can do that, and, you know, they can be in biofilms and such. We've modeled it, and it's really the size of the colony really determines exponentially. There's an exponential decline for the single cell in the colony to become infected, and that really, really, really rapidly declines with the size of the colony. And that's so. I mean, it's super steep. So above 16 cells in a colony, and you're home free. Unless you're infected in the colony, because then the virus is home free, because it has that. But it's rare. Yeah.
Thank you. It was really a new world you opened up to me.
That's
please.
cool. Thank you.
I just wonder, I thought about the previous slide from Penny, where she described there's more phosphorus in the Pacific. Is it fair to say that viral lysis is even more important in the Pacific than the North Atlantic? I'm
not sure whether you can state it's more important, but it could well be that there is a higher viral production, like in the total viruses that are produced. But it could also be that the hosts are growing faster because they are less limited in the phosphorus. Looking at just the phosphorus, something else might limit them. And then the impact of the viruses does not necessarily have to change.
No, okay.
But it's something that would be really cool to check. Yep.
In the back.
Yeah, hello. I was really interested in your measurements of the ratio between the lytic to the grazing rates, because there's been some work out of the Lindau group with modeling by the White's group showing that suggesting anyway that it's a grazing-dominated system in Prochlorococcus at Station Aloha. So I was really interested if you could comment on the methods and maybe their methods and how you think we can resolve this grazing versus lytic debate.
This method uses this principle where, there is, with all the methods that we use so far, there are quite some buts. And that holds for the grazing methods even as well, but it also holds for the viral lysis methods. It's not a simple thing to do. But this particular method is relatively elegant in the sense that what you do is you dilute out the predator effect. So you make different dilutions. You've got the whole water, as we call it, the 100%, and you bring filtrate to it that is taking out the grazers. And now we also use that. We take out the grazers and the viruses. And then the difference, because what happens then is if you take out the grazing pressure, then over 24 hours, because a lot of the algal cells are having a synchronized cell division, so you have to wait the 24 hour at least, you see that these cells have been able to grow because they weren't grazed or infected. And so you calculate the apparent growth rate, you plot it against the dilution factor, and then the slope gives you the potential grazing, and the difference gives you the viral lysis there. So total and grazing. So the method has restrictions because you need to lyse within the 24 hours because we're counting algae. And you don't want to lyse within 12 hours because then you've got a reinfection thing happening. A lot of algal cells actually match that. There are some, especially diatoms, that take longer to lyse. And some EHUX, depending on Emiliania huxleyi, depending on the start of the bloom or when they are in the bloom. But it works pretty well, and all the buts on this particular method underestimate the viral lysis. Why people would say that they match everything with grazing, I think we have to look at specifically when did they measure, where did they measure. I cannot really answer that right away, but what we find is even we done fluorescently labeled prey, for example, and we find the same grazing rates. So at least we know that that part of the methodology is correct. We also done experimental system with Phaeocystis, and we ran the same method for viral lysis, and that matches. So we try to kind of like, prove that we have a good method, but yes, I don't know why specifically. In our case, we actually need always, but as you could see, sometimes, of course, you have some days or some stations where the lysis was just a little bit. If that's the day that you measure, then yes, the grazing will do it. Yeah.
Okay.
Lunchtime.
It looks like it's lunchtime. There's food outside. I think this has been an excellent start to this Microbes Rule the World. And I'd like to thank Penny for her talk, but also congratulate her and thank the speakers and the audience for the stimulating discussion. And we meet back here at 1:20. So thank you.